Hi everybody, welcome here. I'm Weikai Wu from National Taiwan University Hospital, Gastroenterology Department with special interest in gut microbiota. I'm happy to share the content of this interesting academic poster with you here. Also, it is not selected to be the oral presentation by the committee in the first place. This poster highlights the metabolic effect of gut microbiota, focusing on its trimethylamine N-oxide, so-called TMAO producing ability, which is essential on the health condition of cardiovascular system. When we have a big meal, it is not just digested by ourselves. Actually, it was enjoyed by the gut microbiota in our body. And within the big meal, there is a kind of nutrition called carnitine, which is commonly found in the food, especially rich in meat products. When you swallow a piece of steak, the undigested carnitine will be metabolized to a trimethylamine, so-called TMA, by the gut bacteria. The TMA is then absorbed and transported to the liver through portal system and was transformed to TMAO by a group of enzymes called hepatic flammine monooxygenase. The TMAO in the circulation can be harmful to the cardiovascular system, which may lead to a broken heart. Interestingly, researchers also found antibiotics can block the pathway through inhibiting certain gut flora. And our lab also showed that allicin, a natural antibiotic derived from garlic, can elicit similar effect. But is everybody in the population equipped with these bugs to hurt our heart? The answer is not necessary. Researchers have found omnivores and vegetarians have very different TMAO producing ability after giving them a 250 mg isotope L-carnitine capsule, which is so-called carnitine challenge test. In our study, we did not have isotope carnitine capsule. Instead, we include 6 volunteers. 3 are omnivores and 3 are vegetarians, and give them 2-gram L-carnitine tablet without isotope doubling, which we call modified carnitine challenge test, and collect blood samples in the following indicated times. We found the plasma level of TMAO in vegetarians is quite low after carnitine challenge test, but the level in omnivores start to rise sharply at 8 hours after carnitine challenge test. And in the study not shown here, we found the level of TMAO in omnivores can reach much higher at 24th hour. So people may ask, why is the difference of TMAO producing ability between omnivore and vegetarian? And current data using animal model suggests that the carnitine itself can be a trigger of TMAO producing ability. But previous study fed mice with very high dose L-carnitine. Assuming 5 ml per day for a mouse can be equal to 66 pieces of a 5-ounce steak per day for a 60 kilogram adult. And in the real world, we don't consume such high amounts of carnitine from the food, and we are not satisfied with the result to explain difference between omnivore and vegetarian for TMAO producing function. So we conduct another animal study by giving mice much lower carnitine daily intake and shorter duration, and perform carnitine challenge test for the mice thereafter. In the CHO-DIE group, The plasma TMAO level is low and stable, while the carnitine diet group is significantly higher at 2nd, 6th, and 8th hour after carnitine change test. For the stool analysis, using next-generation sequencing, we found the composition of different treatment groups were separated by principal component analysis. Therefore, it suggested that the host with diet of no carnitine supplement tend to absorb carnitine directly without further microbial metabolism, and carnitine itself is good for the health of heart, according to several reports. On the other hand, the host fed with carnitine supplement, despite low daily amount level, will be gaining shorter ability to produce TMAO in the body and hurt the heart. So, it is reasonable to conclude that the different TMAO producing ability is because the diet containing 0.02% carnitine solution for 6 weeks, which is only equal to a 5-ounce steak per day. And this diet difference can shape the function of gut microbiota. In conclusion, we conducted an experiment to prove that dietary level of carnitine intake can induce naive gut microbiota to be a TML producing factory, and it is more reasonable to explain the different cardiometabolic function of gut microbiota between omnivore and vegetarian. Thank you for your watching. If you have any question or you are gut microbiota crazy fans like me, don't hesitate to send me mail for further discussion. And don't forget to give my boss a carbon copy or I may get into trouble. Hope you enjoy it and see you next year. Goodbye.